important variables like FAC is there, MgSO4, K2HO4 and pH. So we got to know that these are the 4 variables, 4 important significant factors which are playing role in enhancing the PBP, and these tools to be monitored through AI based tools and try to develop the CNN based mechanism so that an enhanced production is given. So CNN model, AI model is like a brain, okay. You are putting your information that these are the 4 factors which will give you enhanced production. So if you get a variable mechanism, you try to understand various factors together, then definitely you can run the setup in AI tools, and then a data will be generated with 30 experiment, 20 experiment, whatever you plan, and then you do the wet lab again and try to see whether the AI based generation is correct or not. If not correct, again go RSM, do it again. So it is a process, continuous process. After 2-3 experimentation, you came to know that okay, these are the crucial factors and AI can enhance the production. So I will show you that thing also. So this multi-objective optimization, there is not only this tool, there are so many tools available, okay, but here we have used the multi-objective tool and we generated the algorithm to populate all these input factors, and the best output retains with the especially all these experimentations. So the best outcome means these conditions are the best one, 2 or 3 conditions, and 1 condition we can choose is for the industry. So for industry, why it is used, why for a researcher it is important, the significance is that it is very fast. Second, it is accurate, and third, it will give you the real values based on the various type of other tools which are available. So you are using combinatory tools, you are using fuzzy logic, you are also using ANN, and you are also using genetic algorithm. So this is a combinatory tool which you use for enhanced output. Now these are the regression plots which tells you the value, which we will be showing you in the tutorial also, and this is a connected neural network platform and also the validation check. So because we have to do the validation check also, and then after that we came to know about that. And then this is the regression plot of artificial neural network, and also the optimal production generated through this genetic algorithm has to be validated through wet lab further. So one experiment is not sufficient, you have to try 1, 2, 3, whatever experiment you can plan. Now if you see this paper, I suggest Gaurav, you can get this paper from the lab and give it to them, because this paper gives you the whole process. We have not much time to discuss all the process together, but this paper will give you idea that how these phycobilic proteins can help, can be enhanced using this type of genetic evolutionary algorithm. And when you use this type of platform, we have written a very good review also recently. Mohit, one of your review is very highly cited, you know that, okay. So we wrote that review in 18, I think 2018, 19, and it is cited more than 650 times now. So we have written another review after working for 8 years in AI based platform. We have recently written a small review, and it is published in a book chapter. So you can read that review, and there we have written all these processes, many processes, and the copy is with me. So I will share with you that review, which gives you the all techniques together, because in one paper all techniques you are feeling गर्मी ज्यादा लग रही है, फैन चला दो भाई, उधर वाला नहीं चलता, ये चला दो, इसको बिल्कुल ऐसे ऐसे हो रहा है, वहां बैठ जाओ, वहां ज्यादा है, कुछ लोगों को गर्मी ज्यादा लगती है, हाँ चला दो, ये वाला फैन चला दो, ओके, अभी शायद थोड़ा अच्छा लगेगा, ठीक है. So after this reading this paper, you will be able to understand. And here what we have done, we have taken this CCC 421, and one of my student, I think he worked in IRI also for some time, and then he designed this, and it is also a very good development. And you can see this work we have collaborated with Dr. Deepak Chavda. He is a mechanical engineer. He is not a biotechnology person. Okay, and he helped us to learn the design. He you might be studying in the gene network when Professor Abad will be telling about that. So we have done this GSMM model, and here we collaborated with four people here. Okay, Amit is right. He was earlier in Japan. He was working on plant-based GSMM model, but now he is working with microbial system also. So he was my first second batch BTech student, and he is now faculty in US. So he helped us in understanding the GSMM model, and nowadays he is applying these models in various other applications. So this genome scale metabolic model will also able to understand that why these glycobilic proteins are enhanced. Okay, what is the capability, and do we have any clue where are the crucial genes responsible for enhancing the glycobilic proteins? We are enhancing the process, we are enhancing the production, we are enhancing its amount, but we don't know how we are able to enhance it. Is there any possibility to see at genetic level or at genome level? So that work also we combined here. It was very preliminary, but we developed this hybrid machine learning tools for PPP in Nostock 403, and we got an increase of 61.76%. Okay, so 61% increase in PPP was observed, and 90% in the cell dry mass. So that is also very good. And then potential fluxes, as I told you, that how this PPP production is enhanced, we have to see through the flux, that is the FBA. Okay, that is the flux balance analysis. So that is also very new tool. You can read and understand about it. That is the new tool which is developed. Okay, can we take in the last, you note it down, okay, so that we can finish on time. You just note down your query. All of you please note down your query. We'll take in the last. Now, this is the second case study, as I told you. Second study of the work which uses AI tools. That was PPP production. This is wastewater bioremediation, value addition. Okay, so we, one of my students was working recently. Last year he submitted his thesis. He got awarded, and he was working on wastewater biomediation through microalgae. So he and our collaborator, Professor Prof from IRI, we both discussed about using AI tools for wastewater remediation. So we thought how the FIFO remediation can be enhanced. So he collected lot of wastewater microalgae. And why we are taking up wastewater zone microalgae? Because they are trying to sequester, they are trying to give nutrition.
why excellent growth of microalgae is observed. Okay, though it is toxic also, but it has lot of phosphorus, it has lot of potassium, it has lot of nitrogen, it has other chemicals also, because it is combining all types of plants. So still, if we get with biomass, we can use it for various purpose. Okay, and this is the avenue which you can take up. So our target was isolation. Then we do this microalgae and we see that how high value products can be generated. So this is the sample site. It is in Delhi. Those who are from Delhi, they know it. It is a major sampling site, and Narayana industrial area. You know about it. And this wastewater site was taken, Mayapuri also, and the samples were taken because they are the major sites. And then we did the single factor optimization. Then with this role of salt, that is sodium chloride, and then RSM we have designed, then AI based platform as described earlier, and then we did the multi-objective. Here we have used the MOGA tool, that is the multi-objective genetic algorithm, that is an MOGA tool, and this MOGA tool was very helpful to understand to optimize the production of this nutrient sequestration. So we have done that, and then we did the fatty acid profiling and other aspects which I will be covering in the coming slide. Now if you see, this is the reduction efficiency of different microalgae when cultivated in the wastewater. Why we have chosen microalgae? What microalgae we have chosen? What is the benefit? It is given. Okay, so if you see this TSS, TDS, BOD, all wastewater profiling. Okay, and based on that, we have seen that Chlorella soropiana MS21. This is a very excellent species. Till now, last 4-5 years, I have seen this species is very good. Now, one of our students is also working on biopolymer. Okay, with this species only, and PhD also. So this is a very good microalgae, and probably this has a better capability to produce other product also. We have not seen other products, but we can understand that. And if you see the second level of this removal concentration in agriculture wastewater, this is the industrial wastewater. So we have divided into two categories: agriculture wastewater and industrial wastewater. So these two wastewater, you can see that the specific capability of these algae is removal of various types of pollutants, nutrient sequestration, and recycling is already established. So if you see here, this is the growth profile. See, all your studies, whether it is AI, it is genomics, it is proteomics, omics, all is based on your wet lab map. So if you have the information in wet lab, try dry lab. If you have dry lab information, try to understand through wet lab. So this is mutual. So here, if you see, these are the different microalgae which we have taken, and then we took this specific growth rate of the selected 5. Okay, and then we gone with this MSP1 finally. So you can see here the production. So wastewater can be helpful in production of this biomass, protein, carbohydrate, chlorophyll, carotenoid, lipid, and all that. So these are the important products which are available. They are produced only using the industrial wastewater or agriculture wastewater as a media. We have not used any fancy media, we have not used anything, and these metabolites, these crucial metabolites are used because of the capabilities of the algae. And after using AI tools, I will show you in the coming slide, the production was enhanced. So you are able to get value from waste. So you are using algal system which is already growing there. It is not easy to demonstrate it in the bigger scale because you need a bigger pond, bigger systems. But if large scale is successful, definitely you will be able to have this in the bigger scale as well. So you can see these are the results. You can see in the paper, I will show you that is the Chlorella soropiana MSP1 cultivated in wastewater in response to one factor at a time approach, and these are the crucial factors. And this is the biomolecular lipid E, and these are the surface plots, and then these are the histograms. I am not going in detail because I have already then validation setup, and then Pareto chart, okay, to understand that how much removal is there. Now, after doing this optimization, you got that, okay, the production is fine, everything is giving good rise, and AI tools are effectual. Okay, sometimes if AI tools are not effectual, you are not able to get the good output. It happens. Recently, it happened with us also. Okay, we used RSM, we didn't got good results. When we tried AI tool also, we didn't got good results. So it is not always that AI tool will give you the best one. Okay, sometimes they give you, sometimes they didn't give you. But that is you to decide what are the crucial factors which you want to take help from these tools. So if you are able to decide about significant factors which are controlling the production of the biopigments or metabolites, you can definitely think about. So you can see we did to wet lab to understand that what are specific metabolites, because we are able to get these lipids, we are able to understand that. So we did this, and what is happening inside the cell, because we are biotechnology, microbiology people, so we also understand that what is happening inside the cell. So that AI cannot tell. Okay, you have to go through a scanning electron microscope, you have to see what is happening, how the wastewater remediation is taking place. So we did that to prove that, okay, what is happening, why we got enhanced, because there is lot of activity inside going on. So we have done this study and tried to identify the metabolites. Though we are not able to identify all the metabolites, but we were successful to understand that, okay, these may be the crucial metabolites. And if you see, this is the scanning electron microscope image, you can see that what is happening in the cell. You can see in the picture A, this is freshwater media, and the second is ANN optimized industrial waste wash. So this is also proving that, okay, after this sequestration activity, the cells are also able to change it, and they are in action. Okay, so once they are in action, they are definitely doing something. That's why metabolites are released. That's why nutrient sequestration taking place, and that gives you a profile about some crucial aspects that AI can play a very important role when you talk about the bioprocess design. So this is and then you can see the list of metabolites. We have not worked a single metabolite till now. So we want the student to work on each of these metabolites, because each metabolite is having a different cycle. So my student who was doing this job and he finished this PhD, he has to work on each metabolite. And when you do this specific metabolomics study, you can understand that how each metabolites are controlled and how to enhance these metabolites in further steps. Okay, so these are different metabolites you can see from our data, and these metabolites are very useful. If you Google any of them, you will know they have a quite big role in pharma industry, in medicine, and in well-being products. So these are very important metabolites. Or key companies, they are selling these metabolites very high cost. Okay, and what you are getting these metabolites out of? Wastewater and algae. So you can understand the power of using microbiology, biotechnology, AI tools all together to get all these tools, and including the omics, which Professor Awad is also covering. Okay, so this is various metabolites you can see, and the list is continued, and some of them are not detected. So 29 you can see detected metabolites, and not a single we are able to work till now, because lot of time it will take each metabolites and getting their production efficiency. So now this is the paper, application of ANN MOGA. This paper is also available, you can read it. And here again our collaborator trained us in and designed the machine for the machine learning tools, Deepak Chabraji. And this is Pupa generation, and Pupa has lot of applications. Those who are working on health area or something, they know about the role of Pupa. And again, this is coming to the third application. Two applications we have covered. The third application is about enzymes, industrial enzymes. As I told you in the beginning industry, the worldwide paper giants, they were having the monopoly about that. When vitamin, lactase, and all those enzymes were started production in various plants, so they also wanted some enzyme consortia to improve the paper quality. And sometimes they are using the chemical-based bleaching methods, which is very hazardous. So if they don't use enzyme, they use bleaching. So when they use the chemical-based bleaching methods, they generate lot of pollutants. So why enzyme is important? Because you will be able to have an eco-friendly paper production. And the paper industry will also throw a lot of waste in various fields. So how the waste can be controlled? If you see Delhi nearby, lot of textile industries are there. Okay, lot of paper industries are there. Cardboard industry, अभी Amazon की packing वैकिंग होती है, कितना cardboard use है. Okay, you see every material अभी plastic नहीं use करना, कागज use करना. So for all that paper कहाँ से आएगा? Where recycling is important. So recycling, reusing, enzyme consortia, paper improvement, bio-bleaching. This is called bio-bleaching. One term is bio-leaching, one is bio-bleaching. Bio-leaching probably you might have studied in VARANASI or BSc, okay, where we use microbes for leaching out the minerals. Okay, that is the different thing. But bio-bleaching is this one, using enzymes for bleaching of pulp and paper, and improving the quality. So we use this consortia, we use this mechanism. So understand the three-fold tire mechanism. The mechanism is to have efficient enzyme production, to have efficient process for industry. We are talking about industry, and this is an industrial project. If you see here, industry person is there, Dr. Nishikant Bharadwaj and Puneet Pathak. They both are industry person. It is not only the large scale study, it is industrial collaboration. So this is a industry Avanta Center for Industrial Research, paper mill campus, Yamuna Nagar, Haryana. Okay, so there is a paper mill in Yamuna Nagar. Probably those who are from Haryana, they know. Gaurav, you have visited that place now? Not yet. Okay, so that is the biggest paper mill, and there are other paper mills like in MP also there is a paper mill. So most of the paper mill, it is easy to use the bleaching chemicals. Chemical सस्ता होता है, you use it and throw it, who is going to see? But it is very dangerous. It is dangerous in terms of it is not able to finish. It will go in the runoff, it will go to the agriculture soil, it will go to the groundwater and other places. So we use this enzyme consortia and we improve the quality, and GIAN tools we use for substantial production and improve de-inking. De-inking means improving the quality of the paper. So we use this in this process. So this paper I will give you, all process is almost same. We have used GIAN tools, and the enzymes we have isolated from T. laboriogenesis wax 25. This is a species which we isolated from Faridabad. Okay, so wax 25 is a very good species of Foromic's laboriogenesis, and we isolated it from Faridabad, and it is also having excellent thermostable enzyme. Okay, thermostable enzyme means it can also work at high temperature, 50 plus. So most of the enzymes, they are denaturated, you know that, okay, at high temperature. But this enzyme is a thermostable, it can sustain up to 50, 55 and also sometimes 60. Okay, so when you do this time course of enzyme, you will understand. So this is another case study. This is also a very important area where you can use AI tools, that is the enzymatic enhanced bioremediation. We have not used much tools in bioremediation. If any of you are working on bioremediation, definitely think about using AI tools. And in this review, we have covered all those technologies and also other technologies which can give you better environmental sustainable resources. So then this is Foromic's 25, wax 25. What tools we have used, I will just show you. And these are the crucial factors which we have targeted, that is incubation temperature, initial medium, nitrogen sources, carbon sources, and inoculum this size. And if you see here, this is the much happening after proper training. So you try these tools in your research, even if some of you are working on disease mechanism or disease diagnostic models, that is also used. Cancer biology also it is used. Those who are working like you people are working. Okay, so you can definitely think about that, and these tools are very good, fast, reliable, and they give you good results. So why not you use these tools to understand that and also learn a new process. Okay. Okay, going to this one which I showed you. This is the improved thinking and bio bleaching efficiency of enzyme consortia from WAPS 25 using genetic algorithm and maker based tools. So variety of applications. I am showing you the proof. Okay, all that has resulted in some output, good significant output. And these all papers are very well cited by others. They are cited, some of our papers, I will show you that record also. They are cited, like your paper is cited very well in some bigger journals like Nature Medicine, some papers. Okay, these tools are used. Sometimes I wonder, because the paper is pulp and paper, enzyme efficiency, it is cited by Lancet. The tool are cited, so the paper is cited. So they are using the tools for their disease diagnostics or population modeling or whatever things. So because the tools are quite important. So if you see here, we have seen the improved thinking, and this paper was published in 2022. This is also a good paper, a review, and this is a tutorial paper which I will circulate you. Okay, which is the synthetic biology and biocomputational approaches for microbial endoglucanates towards their innovative application. Mandeep, probably he knows about Mandeep. They both are colleagues in Delhi University. So Mandeep was working with me for his PhD, and now he is in BU, Ansraj College, Botany Department. So last GIAN he came here along with some colleagues, and this GIAN Mohit is here. So we will be interacting together. So this microbial endoglucanase production and its use have not been done at its start. So this review will tell you all these technologies. So we have only discussed about the microbial endoglucanases, but there are so many other enzymes you can use these tools, and we have also used other tools of synthetic biology. So this is process optimization of another case study which we have done. So that was improved thinking. This is the cellulose iron lactic production of the enzyme. So here we have used another species, that is let's see, GGRK4, and we have used RSM and ANN tool. ANN tool we have used. These are the independent variables which we have used, and total 30 experimental sets we have conducted, and these are the control plots showing it. And if you see how it is developed in this table, you can see what is the difference Okay, you can reuse it, design, get the new paper out of it by using these type of technologies. So using the waste paper is also very important. So that's why we have used the office waste, mixed office waste, okay, and photocopier paper also, newspapers also, photocopier machines, paper waste, okay. So all these things are used in this research. This is the one which I was talking about. This here we have documented all the tools. This is a very recent one, and the book was edited by one of our colleagues in enzyme technology. I think you know about him, Professor. He is director right now in ICER Bhopal. So he was writing a book on enzymes only. So we contributed there, and this is a recent, just came up. The online आया नहीं होगा, but I got the tools, so I wanted to show you here. And here we have all these tools covered together, and in a very simple form, so that you can understand. And this is the paper where we have used this. I think I have shown you this. So to close it, our lab, about our lab, you might have met people. So this is my duty to introduce, because most of the work I am presenting here is done by them. I am only the presenter. Okay, the ground force is something different. So they are doing the hard work. Though I have not presented much work of Nandita, Deepali, Sabni, Aditya here, because they are still, Aditya just got one good paper in AI based tools recently, and Nandita also. I have not shown those because of the limited time, but you can share the paper, it is available online. Okay, so she got the production enhancement of xanthopins, carotenoids. Okay, so carotenoid enhancement she has done. And how much fold you got? 6 fold. That means covered. So that is the beauty of using AI tools. Okay, so she just published in BMC Biotechnology. I think 1 month back, 1 or 2 months back. And one more paper by Aditya, who is here, he is using PGPR. Okay, that is a plant growth promoting microbes and their proteins. So we have used those tools and enhanced a very good enhancement of the PGPR characteristics as well. So Swapni is here, she works on cyanobacterial genomics. Probably she is using some of the tools of Gene Network. So probably Professor Azad will, because that is also excellent tool, Gene Network and design. So you will learn it, and she is learning since last 8 months, 10 months. But learning is a long process, so it may take time for you. But once you are get through these tools, you will be able to understand them. Then we have this Gaurav recently, and these are MSc students. And Monis, I already showed you the work. And Bindu right now in South Korea, he worked on enzyme. And Mandeep, I will show you in the next slide. Ritika is there, she is working as a postdoc. I think you interacted with her. She is working on cyanobacterial genomics and other tools, and also working in collaboration with Nandita and Shabni and Gaurav. These are our lab alumni. Shweta, Mohit knows about her, so I need not to tell, but for you, most of the work here I presented was Mandeep, Dinesh, and Vidhu, and along with some of the work which was done by Sino Group. And these are the students who helped to learn. Rajat is there, Ashwini and Dinesh. These are all not biotechnology students, they are mechanical engineers. So they help to learn the tools, and we help to learn biology to them. And then we have this Twinkle is there, then Monica, Sivasan, probiotics. So we have used these tools in variety of areas. Okay, so it is so my personal experience to these tools is not long, it is almost 8 to 10 years. So I noticed that after using these tools, you are quick, you are accurate, and it is well proved in the wet lab experiment. So nobody can question on using these tools. And many groups, they are using these tools, so it is good for using that. These are other lab alumni. Sanjeev Gupta, I have not presented his work here because he worked on therapeutic enzymes. And then Jahangir, plant microbe interaction. Rameshwar and then we collaborated. Punish is in South Korea, Vishal, South Korea, many people in South Korea. Okay. Smith is in MIT. I think those who are from MIT, they know some of MIT sir. Okay. So she is your teacher, right? So then enzyme work, Valinase work, wax is another species. SS was a Smithy species which we isolated from Jharkhand. Then we have Prithvi, Vishal. This is our network, and the network is able to help us to understand, to learn, to exchange the ideas and see how these tools can be effectual. So if you want anywhere, any help, you can always contact our group to understand that. And this network, we recently established 2 more networks, okay, with the GIAN program, so that will also give very good strength to us. And in this work, like Zhengfei Liu from China, then Pan La, and this Yusu Chang from Taiwan, and also the specific groups from this Brazil Marcia. Okay, so these are important, and there are other areas like some of bioremediation, some of the enzymes, some of the probiotics. So 3-4 angles they are focused on. We have another specific network. I have not presented any work here. That is based on using the various type of AI tools for designing the mechanistic details of enzyme consortium. But we have just started working on it. So probably in another 2-3 years, 4 years, or maybe 5 years, we will have something to present. So this is our lab look, you can see, and this is the old picture of our student. You can get here. This is oldest picture. Okay. So I acknowledge our GIAN program for giving opportunity to interact with you. Our foreign faculty, who will be coming soon now. I told him to relax today for 1 hour. So then our head of the department, dean and directors, vice chancellor of BHU, and Center for Bioinformatics and BHU as a whole. And thanks for your questions, listening, and now we will have the discussion on this, and then we go to the tea break, and then after tea break again another session. Okay. So the schedule is not having the tea break as such, because sometime after 1 hour lecture you feel tea break, sometime after 1.5 hour lecture you feel tea break. So tea break is little flexible. We have kept it 11:30. Okay, so now we can discuss. You have written a lot of questions, so don't ask tough questions. Okay, simple questions so that I can answer. Okay, otherwise I have to read again. Okay. Follow simple questions. Okay. You are in which field? Neuroscience. Okay. So as I told you that AI tools are useful in every discipline. Okay. In neuroscience also people they are using especially the gamifications. You know about gamification technology? Okay. So for the neurological disorders, for various type of stress release and all that, gamification techniques are used. So especially when you talk about neurotransmitters, especially talk about the well-being products. So they are all neurological things. So I have not much experience on that, but definitely when you talk about D levels, right? So D levels also we can think about and plan accordingly. But exactly in neurological aspects, what are the crucial factors you are looking for? You can plan it. And what are significant factors coming up for the neurological disorders? So like you, mostly the neurological disorders you have to target. Okay, but these neurological disorders, and you have to especially think about some solution for it. Okay. So for that you have to plan that what are the significant factors contributing to these common neurological disorders, which is mostly patients are coming. Then you can generate some type of matrix and see these are the common factors. Okay, and then you can also design some of the precision medicine. Okay, for especially when you understand that okay, this disorder is quite significant, and these are the factors which are quite significant. So you can suggest to your patients who are coming, okay, and you can guide them that okay, these are the significant factors. You can take care of it and use in the medicine. Second way is that you do the mapping of various neurological disorders and see what are disorders, what are associated factors, and then generate some matrix and try to see the process out of it. And third way is genetic level. You can segregate genes and see the personalized medicine, what can be useful for what type of neurological disorders. So there are a lot of avenues. Okay, and probably Professor Azad has used one because he was on health aspects. He has worked on some health aspect, so he might have some more idea about it. So you plan it. The tools are common. Okay? So like there is a car, anybody can sit. Okay. So if you have a car, you can use it. See whether it is good for you or not. Whether it runs fast, you want a fast running car, you want a slow running car. But 4 people can sit. So the tools can be used for any field. Okay. But neurology, because I am zero in neurology, so I cannot comment. But there are so many studies which we have read, we have seen, we go through that. अभी brain mapping का एक हमने पढ़ा था just, so that is also using AI tools. Okay, ये भी एक type का brain ही है. AI is also brain. Okay, so but this brain is controlled by the various type of commands. Okay, and this is instant. So we are using this because of certain limitations and certain type of decision making capability. So we can use it. Any other discussions or questions? किसने लिखे? किसी ने नहीं लिखे questions? You can use, if you are working on any area, you have any questions, you can definitely ask. Right now we have another 20 minutes time with us before we break. Okay. So what metabolites? Metabolite we have not yet fully characterized. We have seen these and we have partially characterized them. Okay, and the characterization process is simple. Okay, and you can use the metabolomics tools to characterize them through various types of them. Okay, so you can use HRMS or other tools also. Okay, so these metabolites are two-way characterized. One is untargeted, one is targeted. Okay, so if you do untargeted, you don't get all the details there. But if you do targeted metabolomics, phase-wise you target specific metabolite. So characterization of metabolite, one metabolite will take a lot of time. So 29 I showed, I will not able to characterize because time. What? For what? See, if you get this DC mass and HRMS, they quantify and give you. Yeah, LCMS also. So they quantify and give you. There is no need for manual always. These days we have advanced tools. I think we can show you the SAPI facility maybe sometime, maybe tomorrow. There is a very good facility here. I think you might have seen it. SAPI facility is there. Those who are BHU students, they have seen. The Jyoti is there. SAPI facility. So it is having all sophisticated high-end instruments. So you can see the facility, that is a world-class facility here. And it is used by, we have a biomass also. So you can use this facility based on your requirement. And ज्यादा लंबा लगता है। ठीक है, कोई बात नहीं, चाय का ब्रेक कर देते हैं।